introduced Louis Pasteur and this classic experiment that he put together with regards to this flask, and using it to demonstrate that spontaneous generation does not happen. He really proved that you need pre-existing cells to get new cells. You can't just have cells come into existence. This technique here is still used today to keep bacteria out of things that you might want. For instance, a wellhead that might need to have ventilation, where there's an opening here, will have a pipe that comes up and down so that air can still get in, allow for the air to get, but bacteria won't get up into your well. So this technique that Pasteur developed is still used today. But if you remember, basically what he said was that there is bacteria in the air, and that bacteria will get into your media and cause growth. The growth that you see in your media is not because of spontaneous generation. And if you keep the bacteria out, but still let the air in, you won't get any growth. Because some had argued that the air was a vital resource, and that if you let air in, you'll allow spontaneous generation. And he said, well, if I prevent the bacteria from getting there, I won't get any growth, even if I let the air in. He set this up, and sure enough, this stayed sterile for days, for weeks, for months, for years even. It didn't get contaminated until he would tilt it on the end and allow the broth to flow into the net, encountering bacteria and becoming contaminated. And then we also mentioned that even though he had demonstrated this, it was hard to prove his work there for a while, because a lot of scientists were using hay infusions in their labs, or had hay in their labs, and endospores were within these hay, was in this hay, and that endospore would interfere with this test. So it wasn't until John Tyndall came around. John Tyndall demonstrated that these endospores were associated with the hay, and if you take away the hay, you can then reproduce Pasteur's work. And sure enough, spontaneous generation was finally debunked.
So that was debunked by Tyndall, not Pasteur.
Well, Pasteur set the groundwork. And
but technically, because nobody else could reproduce.
No, not nobody else. There was several that could do it if they followed him exactly, but others were, when they used the hay infusion, didn't get the same result. So there was a mixed result there. Some could do it, some couldn't. But Pasteur set the groundwork. Tyndall just demonstrated it was the endospores that were interfering with the people that couldn't reproduce Pasteur's work. And I think that was pretty much all we focused on with regards to the first chapter. Understand the three domains. You have bacteria or eubacteria, you have the archaea, and that you have the eukarya. And then also understand that the eubacteria and the archaea are prokaryotes, okay? And that a prokaryote is an organism that doesn't have a nucleus, doesn't have an organelle. And the eukarya, or the eukaryotes, do have a nucleus, do have organelles, main distinguishing feature between the two. And then we mentioned briefly viruses that first night, and we'll say more about those when we get to that chapter.
I have a question.
Sure.
So we learned that fungi is taxonomically classified, right? What is taxonomical?
What it means is that the fungi are all related. When you look at fungi, they all come from a common ancestor. So when we say that they're taxonomically related, we mean that they're all coming from a common ancestor. And so fungi they share features and characteristics that a common ancestor had. And so they're all part of the same family, if you will, part of the same group. But protozoa and algae, they don't share those same characteristics between all the groups. And so they probably have more than one common ancestor, maybe several ancestors giving rise to those groups of organisms. So protozoa and algae, we will not consider them as taxonomically classified groups. I mean, they're just individual microbes, really. No, there is no relation implied. When we say these are protozoan, it doesn't mean that they're related to each other. When we say these are fungi, it means they're all related to each other.
Then does taxonomical relationship is related to like nucleons, because nucleons carries the DNA information?
Well, it just means that, yeah, they came from what? All organisms have a nucleus. So, I mean, I don't know what you mean by that. They have similar genetic characteristics, similar DNA sequences for sure, and that's why they have the similar traits that they do. And that's why they're grouped together, because they share those genetic traits. Okay, so we moved on to prokaryotes. And if you remember with this one here, this was just more of a review of all the different types of prokaryotes, and we kind of grouped them into common features that they shared. Again, not implying any kind of relationship among the organisms, only saying that they shared these features. And then we broke them down to the anaerobes, the phototrophs, and the aerobes. Very important that you understand the terms in this case here, because that'll tell you what the microbe does. If I say this is an anaerobic chemoorganotroph, you'll be able to tell me that it doesn't require oxygen and it uses an organic food source. Okay, so those are the things that would be great for a multiple choice question. So we know that anaerobes can be chemolithotrophs, or they could be chemoorganotrophs. Same is true for the aerobes. You can have lithotrophs or you can have organotrophs. And then if you remember the phototrophs, we broke those down into the anoxygenic phototrophs that did not produce any oxygen, and the oxygenic phototrophs. So I would expect you to be familiar with this terminology here, but then also to provide me an example for each of the groups. So, except for, I don't think we really went into this purple bacteria. I wouldn't expect you to provide me a sample here, but the rest of the groups, you should be able to provide me a microbe that would fall within that group.
So for instance, like a phototroph, an oxygenic phototroph, would you be looking for something like a cyanobacteria?
Yes, exactly. And then imagine a multiple choice question where I would say, the following organism is an oxygenic phototroph. And then I would give you 4 choices. And you'd have to identify cyanobacteria as the right choice. Okay? That's kind of questions will be, not all of them will be that easy, but I'm just saying. That's kind of what it will be like. Understand facultative anaerobe. You know what it is. It's an aerobe, prefers an aerobic environment, but can grow when oxygen is not present. Can grow in an anaerobic setting. With regards to the ecophysiology we talked about, we mentioned that the terrestrial organisms have some adaptations. The endospores was the main one that we talked about. And we also talked about a plant association, rhizobia, and the fact that it is associated with root nodules of legumes and its feature, its characteristic is nitrogen fixation. Okay, so going back to one of those vital activities that's carried out by the microbe here. One of I think the best ways to study this particular chapter is to do just this, to kind of set yourself out a flow chart. So you started with the 3 that we broke it down to, and then you break that out further. And remember my telling you earlier in the semester that we can really only remember, between 5 and 9 things at a time. 5 to 9 pieces of information. It's generally about 7 for the average person. So if you break it up, so that this is one chunk of information that you have here, and then you do just the anaerobe, so that's one chunk of information, and then this is one chunk of information. So that you're remembering those, you're actually, if you think about it, and studies have shown this, doing that now gives you 4 separate chunks of information that you can remember, because now it's only 4 chunks of information instead of 17 chunks of information. So group your stuff together when you're studying this material. Create flowcharts and stuff. Problem with flashcards, and some of you probably do well with flashcards, but the problem with flashcards is they don't always give you the whole picture. You're learning terms, but you're not necessarily learning relationship. And sometimes the relationship helps you remember the material. So I would encourage you to draw stuff out whenever you can. Just write it, draw it, whatever you use. So that brings us then to the protozoa. Where did I put those guys? And if you remember with the protozoa, well actually the eukaryotes I guess, we had the 3 groups. We had the algae, we had the protozoa, we had the fungi. I would expect that you could distinguish the 3 groups from each other. What are the main characteristics of algae? Photosynthetic, cell wall made of cellulose. What are the characteristics of protozoa? They lack photosynthesis. They have mode of locomotion. And then we could talk about that mode of locomotion, how